There are volumes of books, articles, and documentaries on the brain. We know so much about it, and yet so little. It seems that the more we learn about this organ, the more we marvel at its function. It is a remarkable control center that has the ability to perceive, analyze and react. It is the site of our personality, intelligence and creativity, and our recognition of God. It doesn't look like much from the gross anatomical point of view, but it is absolutely amazing. In this presentation, we will look at basic brain function and anatomy. We will learn how the brain is protected and isolated from the rest of the body, and yet communicates with it. The central nervous system includes the brain and the spinal cord and technically, the retina as well. This set of notes will focus on the brain. The average human brain weighs about 3 pounds. What the brain lacks in appearance, it more than makes up for in complexity. For this reason, the brain is discussed in terms of 4 major regions, the cerebral hemispheres, diencephalon, brainstem, and cerebellum. This is an excellent summary slide of the brain which shows the structure of the major brain regions and some of their important subdivisions, and indicates what the overall general function of each area is. Read over this slide to become familiar with the major actions and location of each of the regions shown in the picture. The CNS first appears in embryological development as a hollow neural tube which lies in the dorsal median plan or long axis of the body. During week 4 of embryonic development, 3 areas at the anterior end of the neural tube begin to expand. This formed the primary brain vesicles called the prosencephalon of forebrain, mesencephalon or midbrain and rhombencephalon or hindbrain. Development of the brain proceeds extremely rapidly. Just 1 week later, week 5, secondary brain vesicles have formed. These will develop into the main structures of the adult brain. The prosencephalon divides forming the telencephalon which will become the cerebrum, and the diencephalon which will include the thalamus, hypothalamus, epithalamus and retina. The mesencephalon does not subdivide and become the midbrain. The rhombencephalon subdivides forming the metencephalon which becomes the pons of the brain stem and the cerebellum. The myelencephalon becomes the medulla oblongata. This diagram shows the progression of development of the human brain from 5 weeks of gestation to birth. Notice how the cerebral hemispheres overgrow both the diencephalon and most of the brain stem. Also notice that as development proceeds, elevations called gyri singular gyrus and grooves called sulci singular sulcus increase in number and size. These foldings increase the surface area of the brain so we can pack more neurons into a small area. Each cerebral hemisphere is divided into 4 lobes which are named based on the bones of the cranium that surround each of them. The top picture is a superior view of the brain showing the frontal, parietal and occipital lobes. The lateral view shows the temporal lobe. The cerebrum is the most highly developed in humans compared to other mammals probably because we wrote the anatomy books. It is the area of the brain that is involved with complex functions involving many neurons for multiple areas of the brain such as intelligence, creativity and learning. The picture in the slide is of a function MRI of the brain taken while the volunteer was speaking. The brighter the area lights up, the greater the metabolic activity taking place in that region. This is one way that neuroscientists have figured out what part of the brain is predominantly involved with what function. The cerebrum is made up of 3 major components, the cerebral cortex, white matter, and the basal nuclei. The outer cerebral cortex covers the surface of the cerebrum and is composed of gray matter 2 to 4 mm thick. It is the folds of gray matter that comprise the gyri. Sulci sulcus are the grooves between the gyri. The gray matter is made up of neuronal cell bodies dendrites, associated glia and blood vessels but no fiber tracts. Different areas of the cortex contain neurons that make up motor, sensory and association areas. The cerebral cortex contains billions of neurons arranged in 6 layers. Different types of information is processed in different regions of the cerebral cortex. Even though the cortex is only 2 to 4 mm 1/8 of an inch thick, it accounts for about 40% of brain mass because all the convolutions triple its surface area. The white matter consists of myelinated fiber tracts which carry specific types of information between different areas of the brain, and connect higher and lower divisions of the CNS. Deep to the white matter are islands of gray matter called the basal nuclei. These function to regulate voluntary muscle movements. Damage to the basal nuclei result in some of the most devastating diseases affecting the brain. As mentioned in the previous slide, The sulci sulcus are grooves between the gyri. There are some less numerous, but deeper groove called fissures which separate large regions of the brain. The longitudinal fissure is a single, deep fissure that separates the hemispheres. The falx cerebri is an extension of the dura mater that runs between the hemispheres in the longitudinal fissure. The cortex is divided into functional areas which are numbered and mapped out on this diagram. All functional areas fall into one of three functions: motor, sensory or association. In this picture, notice the deep central sulcus that separates the frontal and parietal lobes of the cerebrum. Anterior to the central sulcus is the precentral gyrus, which is involved with primary somatomotor commands voluntary to skeletal muscles. The postcentral gyrus, seen as dark blue on the diagram, is involved with receiving somatic sensory stimuli. Notice that behind the postcentral gyrus is a region colored light blue. This is the somatosensory cortex. This is where we interpret and store sensory information. For example, what a paperclip feels like, the information is stored in the sensory association cortex. From this point on, we can know what an object like a paperclip is without even looking at it. The phenomenon of phantom pain also has to do with sensory information stored in this area. The dark blue of the occipital lobe represents the primary visual cortex, and the light blue is the visual association area. When you read the word feather, you instantly retrieve an image of a feather stored in your visual association area. There is also a primary auditory cortex for hearing and an auditory association region, called pharynx, that is critical for normal speech. In the frontal lobe, there is an area called Broca's motor speech area, also critical for speech. The frontal eye field is a motor area that helps program the eye muscles, and the premotor cortex is involved with programming simultaneous or sequential movements as when doing a learned dance, playing piano, or texting. This slide shows a diagram called the homunculus. The areas of the body for both incoming sensory information and outgoing motor information has been mapped to specific sites on the primary motor precentral gyrus and primary sensory postcentral gyrus. Notice that the relative size of the body part has nothing to do with its spatial representation on the cortex. For example, the trunk is a large proportion of our body, but is represented on the precentral gyrus by an area of cortex much smaller than that of the thumb or lips. This is because areas where we need either fine motor control, or special tactile sensitivity like our fingers require more neurons than an area where there is little need for precise movements or sensation. If we were to draw the human body according to the proportions represented on the precentral gyrus cortex, we'd have a very odd-looking individual. Notice that the lips are bigger than the hips. The hands are huge compared to the trunk. The tongue is a small organ but contains many intrinsic muscles that are critical for important functions such as speech, manipulating food, and swallowing. It also is laced with tactile receptors and taste buds. As James so aptly put it, so the tongue also is a little member, and boasteth great things. Behold, how much wood is kindled by how small a fire. Jam 3-5. Definitely, my tongue has gotten me into trouble. The tiny tongue can be used for edification and for proclamation, and that every tongue should confess that Jesus Christ is Lord, to the glory of God the Father. The white matter of the cerebrum lies underneath the gray matter of the cerebral cortex. It consists of mainly myelinated axons comprising fiber tracts of different types, such as commissures, association fibers, projection fibers, and the internal capsule. Commissures connect the corresponding gyri of the two hemispheres. This allows for the right hand to know what the left hand is doing. Association fibers are connections between the gyri of the same hemisphere. Projection fibers collectively form the internal capsule of white matter. Projection fibers consist of ascending and descending tracts that transmit impulses from the cerebral cortex to spinal cord and from spinal cord to cerebral cortex. All projection fibers pass through the diencephalon. The third component of the cerebrum are the basal nuclei. They lie deep within the cerebral white matter. Nuclei are groups of neuron cell bodies in the CNS. The basal nuclei are functionally associated with other lower areas of the brain such as the subthalamic nuclei and the substantia nigra of the midbrain. The basal ganglia play a role in large automatic muscle movements, such as swinging the arms while walking, regulation of muscle tone, and inhibition of unnecessary movements. Lesions damage to the basal nuclei lead to devastating impairments of movement such as tremor, Valismus and hemivalismus, which are sudden, powerful, uncontrolled movements affecting the entire or one side of the body, and various forms of paralysis and muscle rigidity. The link above shows an example of people suffering from Huntington's chorea. The primary motor and sensory areas are bilaterally symmetrical. We use both cerebral hemispheres for almost every activity. The association areas are not bilaterally symmetrical. Consequently, each brain hemisphere controls a unique set of functions different from the other lateralization. The primary motor and sensory areas are bilaterally symmetrical, they just control opposite sides of the body. The association areas are not bilaterally symmetrical. Consequently, each brain hemisphere controls a unique set of functions different from the other. This is called lateralization. Notice that speech, writing, language and mathematical calculation activities are located in the left hemisphere, and analysis by touch, spatial visualization and analysis is located in the right. We use both cerebral hemispheres for almost every activity, and it is the corpus callosum that connects the special activities performed in each hemisphere to one another. One of the hemispheres will dominate each task. In most people, about 90%, the left hemisphere has greater control over language abilities, math, and logic. For example, the left hemisphere is dominant and working when we compose a sentence, balance a checkbook, memorize a list of anatomical terms, etc. The right hemisphere is involved in visual spatial skills, intuition, emotion, artistic and musical skills. Some people with certain personality traits are described as right or left hemisphere dominant. The next major brain region we will discuss is the diencephalon. The diencephalon sits on top of the brainstem and is surrounded by the cerebral hemispheres. It consists of several major structures, the thalamus, hypothalamus, epithalamus and the subthalamus. We will begin with the thalamus. The thalamus comes from a word that means inner room. It makes up 80% of the diencephalon, and consists of a bilateral, egg-shaped collection of many nuclei. Typically, the nuclei are connected at the midline by an interthalamic adhesion or intermediate mass. The thalamus is a major message relay station. It sends and receives motor and sensory information to and from specific regions of the cerebrum, as well as the spinal cord, brainstem and numerous regions of the cortex. Afferent impulses from all senses and all parts of the body converge on the thalamus and synapse with neurons in at least one of its nuclei. For example, the medial geniculate nuclei receive information from the hearing apparatus called the cochlea, and relays it to the part of the temporal lobe where sound is sorted out. Similarly, the lateral geniculate nuclei receives and relays information pertaining to vision. The ventral posterior nuclei have regions that are involved with the sense of taste, as well as regions involved with relaying crude somatic sensations such as touch, pressure, vibration, heat, cold and pain. Crude means that at this level, these sensations are recognized only as pleasant or unpleasant until they are relayed to appropriate interpretive areas of the cerebral cortex. In short, the thalamus sorts and edits incoming sensory information and relays impulses having to do with similar functions as a group of fibrous axons to the appropriate area of sensory cortex and association areas. The term hypothalamus literally means under the thalamus. This is a tiny but mighty region of the brain. When viewed from a dissected brain, it looks very insignificant. However, it is an important autonomic nervous system center. It plays a role in regulation of body temperature, water balance, metabolism, and is also the center for many drives and emotions. The 12 nuclei comprising the hypothalamus are separated into different regions. For example, the mammillary bodies are a relay station for reflexes related to olfaction, and they control reflex movements associated with eating, such as licking, swallowing and chewing. The tuberal region contains the infundibulum or stalk of the pituitary gland, and the median eminence, which contains the neurons that secrete hormones that regulate the secretions of the pituitary gland. The supraoptic region lies above the optic chiasm and contains several nuclei including the paraventricular nucleus, which is the source of oxytocin, and the supraoptic nucleus, which synthesizes antidiuretic hormone ADH. We will learn about these hormones when we study the endocrine system, but suffice it to say, oxytocin is known as the love hormone. It is also involved in labor contractions and the milk letdown reflex. ADH regulates blood pressure and body fluid compartment balance. One of the most interesting G regions of the hypothalamus is the preoptic region. The nuclei here regulate autonomic functions. They receive sensory information from internal and external environments, and regulate visceral activities, such as heart rate, peristalsis, contraction of the urinary bladder, and body temperature. It is here that we find nuclei that regulate food intake via feeding and satiety centers, and also thirst, waking and sleep, and primitive emotions such as rage and aggression. Hypothalamic disorders can be the underlying cause of numerous abnormal conditions such as severe body wasting, obesity, sleep disturbances, dehydration, and a wide range of emotional imbalances. It is also thought that if the hypothalamus doesn't receive the normal sensory input during early development, it fails to function correctly. For example, if infants are deprived of parental care and nurturing, signs of a syndrome known as failure to thrive becomes obvious. Failure to thrive is a clinical term describing a delay in a child's growth or development. The epithalamus is the most dorsal portion of the diencephalon. It forms the roof of the third ventricle, one of several cavities in the brain for the production and circulation of cerebrospinal fluid or CSF. The most important component of the epithalamus is the pineal gland. The pineal is both a brain structure and an endocrine organ because it secretes the hormone melatonin. Melatonin makes us sleepy at night and helps to regulate daily rhythms. The levels of melatonin are regulated by light levels. At night, melatonin levels are high and we feel sleepy, however light inhibits melatonin release and we feel more awake. The brainstem is about 3 inches long, and the diameter of a thumb. It is evolutionarily the earliest part of the brain to develop, and so the brainstem of different kinds of vertebrates functions pretty much the same. It is possible to live in a vegetative state with severe damage to the cerebral cortex, but damage to the brainstem is life-threatening. The brainstem is involved with regulating vital activities such as breathing, heart rate, blood pressure and many more. It is also involved with regulating movement because of its connection to the basal nuclei. The brainstem consists of 3 parts, the midbrain, pons and medulla oblongata. The midbrain is a relatively small part of the brainstem. It contains several important structures such as fiber tracts and nuclei. The tracts include the cerebral peduncles which connect the lower brain with the upper brain or diencephalon. Peduncle, by the way, means little feet, aptly named because the cerebral peduncles support and connect the cerebrum to the midbrain. The peduncles contain motor fibers from the cerebral cortex going to the pons, medulla oblongata and spinal cord such as the pyramidal corticospinal motor tract. The sensory fibers come up from the spinal cord, through the medulla, pons, midbrain and then to the thalamus of the diencephalon. There are also a pair of superior cerebellar peduncles which connect the midbrain to the cerebellum dorsally. A tiny canal called the cerebral aqueduct travels through the midbrain, and connects the third ventricle to the fourth ventricle. One of the most obvious external features of the midbrain are 4 rounded bumps collectively referred to as the corpora quadrigemina, so named because they reminded some anatomist of 2 pairs of twins, like Gemini the constellation. The twins of the corpora quadrigemina are the right and left, superior and inferior colliculi. These structures are involved with programming eye, head and neck movements so that we can track or locate a visual or auditory stimulus, respectively. Notice the large orange structure in the diagram. This is the end of a collection of nuclei and tracts known as the reticular formation that extend through the entire length of the brainstem. It is involved with visceral function, and contains a special group of neurons called the reticular activating system or RAS. RAS plays a role in consciousness, states of awareness and sleep/wake cycles. Damage to RAS can result in permanent coma. 2 of the 12 cranial nerves arise from nuclei located in the midbrain. Cranial nerve 3, the oculomotor, innervates most of the extrinsic eye muscles we function to move the eye. It also contains some parasympathetic fibers that control the intrinsic smooth muscle of the eye which change the size of the pupil and help the lens bulge for near vision. The nucleus of cranial nerve 4, the trochlear nerve, is found laterally in the midbrain. This nerve innervates but a single eye muscle. Notice also that there are several obvious nuclei which were named because of their color. The red nucleus gets its name from its rich blood supply and because its neurons contain iron-containing pigments. The red nucleus consists of cell bodies whose axons comprise the descending rubrospinal tracts for subconscious regulation of upper limb position and movement. The substantia nigra contains large, darkly pigmented neurons that produce the neurotransmitter dopamine. Death of the neuron cell bodies of the substantia nigra result in Parkinson's disease. The substantia nigra and the basal nuclei of the cerebrum are functionally related. Recall that the basal nuclei help regulate voluntary motor activities by modifying instructions sent to the skeletal muscles by the primary motor cortex. The dopamine produced by the neurons of the substantia nigra tone down the output from the basal nuclei. Parkinson's disease results from the death of the cells in the substantia nigra. The decreased levels of dopamine leaves the action of the other stimulatory neurotransmitters unopposed, resulting in tremor, muscular rigidity, and decreasing range of movement. The term, pons means bridge because the pons connects different parts of the brain with each other and with the spinal cord. It also connects to the cerebellum via the middle cerebellar peduncle, and it connects the medulla oblongata with the midbrain. Although the pons is mainly fiber tracts that link up different parts of the brain, it does contain gray matter such as the nuclei of cranial nerves V7, and contains a nucleus which makes up part of the respiratory center which controls breathing. The picture on the right is of a section through the pons. It shows the massive cerebellar peduncle, and the crossing fibers of various ascending and descending tracts. The diagram on the left shows the nucleus of the trigeminal nerve and some of the pontine nuclei involved with autonomic functions. The inferior-most part of the brainstem is the medulla oblongata. It is essentially a continuation of the spinal cord with which it merges. The medulla oblongata contains nuclei and all of the ascending and descending tracts for impulses leaving the brain and going to the periphery, and sensory information coming from the periphery and going to the brain. Although there aren't any obvious changes in structure defining the end of the medulla oblongata and the beginning of the spinal cord, there are several features that can be seen with the naked eye. There are two bulges on the ventral side of the medulla oblongata called the pyramids. The pyramids are formed from the massive corticospinal tracts which originate from the pyramidal neurons of the motor cortex, thus the name pyramids. If examined with a magnifying glass, it is possible to see where the fibers cross over to the opposite side of the medulla oblongata before descending into the spinal cord. Crossing of fibers, in gene RAL, is called a decussation. The olives, or olivary nuclei are relay stations from midbrain and cerebral cortex to cerebellum. Solitary nucleus integrates and relays visceral sensory information to autonomic centers. On the dorsal surface, the nucleus gracilis and cuneatus relay somatic sensory information to thalamus. Other nuclei comprise reflex centers that control visceral or autonomic functions such as breathing, swallowing, coughing, sneezing, heartbeat and blood vessel diameter. Because many of the functions that keep us alive are centered in the medulla oblongata, injury to this region can result in death. The medulla oblongata also communicates with the cerebellum via the inferior cerebellar peduncles. These fiber tracts carry information from the spinal cord regarding position of joints, length of muscles, state of stretch of muscles and joints, and sends back information to the body to coordinate how much to move what muscles to maintain balance and posture. The nuclei of cranial nerves 8-12 also originate from the medulla oblongata. CN8, vestibulocochlear 4 nuclei total involved with hearing and balance, CN9, glossopharyngeal, coordinate swallowing and salivation, and brings taste fibers from the back of the tongue, CNX, vagus controls the activities of the thoracic and abdominal viscera, CN11, accessory controls voluntary head and shoulder movements, and CN12, hypoglossal control voluntary tongue movements. Many of the tracts cross from one side of the nervous system to the other. The point where they cross is called a decussation. It is important to know where tracts cross to diagnose location of injury site. Fibers originating on the left side of the brain activate muscles on the right side of the body. The nucleus gracilis and cuneatus receive sensory information from ascending tracts of the spinal cord and relay it to the thalamus on the opposite side. Similarly, the pyramids ventral, motor decussate conveying outgoing motor information coming from the cerebral motor cortex to the muscles on the opposite side of the body. The cerebellum or little cerebrum lies in the posterior cranial fossa and projects dorsally from the occipital lobe. It is separated from the cerebral hemispheres by a transverse fissure in which lies a shelf of dura mater known as the tentorium cerebelli. The main function of the cerebellum is to help us maintain balance and posture, and to coordinate, adjust and smooth movement. It integrates sensory information from the muscles, proprioceptors position sense of joints and muscles, visual information from the eyes, and vestibular input from the inner ear. It tells the motor areas of the cerebral cortex what adjustments to make to maintain balance or posture, and is important for skilled, coordinated movements and agility as when dancing, playing baseball, or walking a straight line. Like the cerebrum, the cerebellum has two hemispheres, a convoluted surface and an outer cortex made up of gray matter. The inner white matter is arranged like a tree with many branches, and so is named the arbor vitae or tree of life. Notice that there is a crude outline of the body on the two hemispheres that is analogous to the homunculus of the cerebrum. The histology of the cerebellar cortex is highly ordered. The outermost layer is called the molecular layer. Separating the molecular layer from the inner granular layer is a row of large cells called Purkinje cells which play a critical role in integration of sensory and motor information. If the cerebellum is damaged, movements become clumsy and disorganized, a condition called ataxia, which literally means without order. People suffering from ataxia appear to be drunk because they cannot keep their balance, and can't touch their finger to their nose with their eyes closed. Even without injury, neurons of the cerebellum die as we age. This is why older people have a more difficult time keeping their balance and fall more often. The CNS is soft and fragile, and once neurons of the CNS are destroyed, they cannot be replaced. Therefore, the brain and spinal cord are enclosed in the bony dorsal body cavity, surrounded by connective tissue membranes called the meninges, and float in a watery cushion of cerebrospinal fluid or CSF. There are three layers of meninges. From outer to inner, they are the dura mater or tough mother, the arachnoid mater or spider mater, and the pia mater or delicate/gentle mother. The dura mater is the outermost and strongest of the brain coverings. The cranial dura is double-layered. In the cranium, the outer layer is fused with the skull bones and actually functions as a periosteum. The inner layer is called the meningeal layer and forms the outermost covering of the brain, and continues as the dura mater of the spinal cord. The dural layers are fused together except in three areas where they separate to enclose dural sinuses for venous drainage of the brain and where CSF is. Return to the blood. In several places the inner dural layer forms folds that attach the brain to the cranial cavity and form partitions that limit brain movement. The major partitions are the falx cerebri, which separate the cerebral hemispheres, and the falx cerebelli or tentorium cerebelli that separate the cerebellum from the cerebrum. The middle meninx is called the arachnoid mater because it has spider web-like extensions that secure it to the underlying pia mater. The space between the arachnoid mater and the pia mater is called the subarachnoid space, and it contains cerebrospinal fluid. The pia mater is thin, transparent and adheres snugly to the surface of the brain following the gyri and sulci. The pia mater carries blood vessels that supply or drain the brain. Cerebral spinal fluid, or CSF, is a filtrate of blood plasma, however it contains less protein, more vitamin C, and has a different ion composition. CSF nourishes the brain and spinal cord. It exchanges nutrients and waste products of neuron metabolism. The CSF protects the central nervous system against injury by forming a fluid-filled sac that suspends the brain and spinal cord from outside and inside. It maintains the right ionic balance for neuronal signaling to work properly. CSF is found in the ventricles of the brain where it is produced, central canal of the spinal cord, and the subarachnoid space. There are four cavities called ventricles that lie within the brain. Two lateral ventricles, one in each hemisphere, a central third ventricle by the hypothalamus and inferior and median to the lateral ventricles, and a fourth ventricle between the brainstem and cerebellum. The third and fourth ventricles are connected by a canal called the cerebral aqueduct of Sylvius or the mesencephalic aqueduct. The CSF is secreted by the choroid plexuses found in the lateral and fourth ventricles. The choroid plexuses contain special networks of capillaries covered by ependymal cells. The ependymal cells allow only certain materials to pass from the plasma and enter the CSF. You may recall from our discussion of the neuroglia of the CNS, that ependymal cells line the ventricles and the central canal of the spinal cord. The beating of their cilia circulate the CSF throughout these structures, and into the subarachnoid space. CSF is continuously produced at a rate of about 480 milliliters/day. Obviously, the CSF has to go somewhere after it finishes circulating around the CNS. In fact, it is reabsorbed back into the blood vascular system. It drains into the superior sagittal sinus formed as a space between the two layers of dura mater. Extensions of the arachnoid mater called the arachnoid villi project into the superior sagittal sinus. It is from these mushroom-shaped projections that CSF is returned to the bloodstream. Any condition, disease, birth defect or injury that causes a blockage in the drainage system can result in a condition called hydrocephalus or water on the brain. In infants, the buildup of CSF is interpreted as a growing brain. Recall that the cranial bones have not fused to form sutures to allow the brain to grow. Therefore, if drainage of CSF is blocked, the cranial bones continue to grow, resulting in a disproportionately enlarged head. The situation is dangerous in adults because the sutures have formed and so the brain is squeezed against the hard skull bones. This can result in severe brain damage, coma and death. Hydrocephalus is treated by making a shunt from the lateral ventricle to the superior vena cava and or subclavian vein to allow the CSF to drain. As we age, tissues of different kinds sometimes calcify. This happens to the arachnoid villi. Once they become impregnated with calcium, they become hard and are known as arachnoid granulations. On the inside of the cavarium, the arachnoid granulations erode pits into the skull bones on either side of the superior sagittal sinus. Paradoxically, the brain is one of the most metabolically active organs in the body, and yet there is no direct exchange of materials between the blood and brain. The brain requires glucose to make ATP. It consumes 20% of the body's oxygen supply in a resting adult, and even more when thinking or performing a difficult task. However, no other body organ is so absolutely dependent on a constant internal environment as the brain. If the brain was exposed to even slight chemical changes, uncontrolled neural activity might result. Consequently, neurons are kept separated from blood-borne substances by the blood-brain barrier. The barrier is made up of a leak-proof type of capillary called continuous capillaries. Ependymal cells help to form the barrier, and astrocytes help to regulate what can pass to the brain. Large molecules such as antibodies, plasma proteins and pathogens are prevented from contacting the brain. Fluctuating hormone and electrolyte levels are also held in check. Glucose and water can be exchanged, but this is highly selective and directional. However, respiratory gases oxygen and carbon dioxide and fat-soluble substances can diffuse through the barrier. This explains why blood-borne alcohol, nicotine, certain drugs and anesthetics can affect the brain. There are a couple of areas in the brain where the barrier is a little leaky, but this serves an important functional purpose. Recall that certain areas of the brain functionally overlap with the endocrine system by synthesizing hormones. Hormones are carried in the bloodstream to their target cells. In order for the hormones produced by brain structures to gain access to the bloodstream, there are selected parts of the blood-brain barrier that allow them to pass.